Okay, thanks for the introduction and the opportunity to talk today. Um, what I'm going to talk about is work that we're doing for a project that's funded by AgriFutures Chicken Meat. We're a bit over halfway through the project, so it's still very much a work in progress. So really what I want to tell you about today is why we think it's worth doing and our approach to sort of tackling the issues as we see them. So why use probiotics? I'd just like to sort of quickly run through that, why we are actually interested in generating a sort of new generation of probiotic products. With the move away from antimicrobial growth promoters, there's a real desire to come up with other means to try and maintain general health, but gut health in particular, and to sort of increase or maintain at least productivity. So that's sort of always been one of the main drivers, to find alternatives to AGPs that will support health and productivity. And now there are many products already out there in the market that people are using. These products have different penetrations in the market, but there's a long list of them. So there's prebiotics. So these are the sort of complex carbohydrates that basically feed the gut microbiota and can lead to the production of really useful metabolites like butyrate. The probiotics that I'll talk about today. There's also postbiotics. You may be a little less familiar with things that are called postbiotics, but they're basically fermentation supernatants. So it's what's there after you've grown a bacteria. There can be very useful metabolites in those, and use that sort of product. There are a lot of phytobiotics out on the market now. So these include things like essential oils from plants. They're getting quite a high level of acceptance these days. And then another sort of big area are the short and medium chain fatty acids, protected butyrate products, things like that. There's also a range of yeast products. These really fall into some of these other categories as well. And more rarely, there are some people using algal products around the world. So they're the things that we can use to address this issue of, the reduction of use of AGPs to try and maintain health and productivity. But what I'm interested in, I'll tell you about today, are the probiotics. So what exactly are probiotics? The sort of formal definition that's most accepted these days is that they're classified as live microorganisms, which when administered in adequate amounts confer a health benefit on the host. So the really important thing is live microorganisms. So it's not just adding the culture contents or whatever, the metabolites that might be in the culture. It's the fact that the organisms are live and function partly because of that. So the action of probiotics can be sort of wide and quite varied. What people sometimes are a bit sort of confused about is that probiotics are a vast range of different products. So each probiotic is different. It's a different strain of bacteria that has different properties and they can work in different ways. So no one probiotic is likely to have the potential to activate all these mechanisms of action. They're likely to have 2 or 3 of these maybe. So in terms of mechanism of action, improving gut health is a very broad thing, and it's really made up of all the things beneath it. So the top 2 are probably some of the most interesting ones these days. First is to improve gut integrity. So this is to make sure that the gut barrier function is maintained and there's not leakage of bacteria systemically into the bird, causing infections. Some probiotics have a really strong activity in maintaining that gut integrity, that barrier function of the gut. Some of them also stimulate the immune system. You know, some phytobiotics do this as well, to make the immune system stronger and to perhaps suppress some aspects of the immune system as well as advancing other aspects of it. Some of the probiotics express antimicrobial proteins that can directly kill pathogens such as Clostridium perfringens. So a lot of probiotics are sold on the idea that they can directly kill a pathogen such as Clostridium perfringens. But others have a more subtle effect on pathogens, and they can actually sort of take up an ecological niche within the gut, so there's no binding sites for pathogens when they go in there. So it's more by excluding the pathogens rather than killing them that they may work. Probiotics can also just improve the gut microbiota, so the structure of the gut microbiota, to give you a more healthy overall gut microbiota that functions better. Importantly, some probiotics directly increase short-chain fatty acid production, use really important things like butyrate, which is a really significant energy source for the gut. Some can also produce digestive enzymes that can help in recovery of nutrients. A really weird one, which there's no probiotics for chickens out there that I know of that actually do this, but it's becoming more of a thing in human probiotics, and that's that probiotics can actually be used to modify mood and behavior. There's this gut-brain axis which can be activated, and perhaps in the future there may be some application in animals to do this sort of thing. So that's the sort of thing that we're hoping that a probiotic will do. And as I say, one probiotic won't do all of these things. It'll only do a limited range of those. But what are why do we need more probiotics or different probiotics? There are already a lot on the market. Some of them are getting very good acceptance. People seem to be getting good results from them. Do we need to look for more of them? And here are the issues as I see them. There's a very variable amount of data to support the use of probiotics. So a few of the products out there have some really good data. There's a lot of products that are available that really have no supporting data. And the experience out there, sort of anecdotally and a little bit in the literature, is that the performance of an individual, any one probiotic, can, for some of them, be quite variable. So sometimes they seem to give really good results, and at other times you don't seem to get much of a response using them. And one of the other big things for all the products that are out there at the moment really is this need for continuous dosing. And that's to some extent a bit of a puzzle, I think, because if the key is that these things are live organisms, why do you have to keep dosing them if they're working effectively? I mean, if you put them in there and they become part of the gut microbiota because they're living, then why is there this need for continuous dosing? I think there's something there that's not quite right, that can be improved in future products. So, why do some of the probiotics that are in the literature and used in the marketplace have these sort of issues? One of the things is that, the bacterial strains that we're using in the poultry industry, not all of them have actually come from chickens. So some of them may be from environmental sources, some of them may be from other animals. So there's this sort of attempt to have a sort of one-size-fits-all approach with one particular probiotic being used in multiple species, pigs, chicken, cattle. And is that a reasonable expectation? I mean, for some special strains that may work, but for others, perhaps because they're not actually tailored for use in chickens, we have this need for continuous dosing. This is not a really, this is why in practice out in the field, sometimes you get variable results because the products that are actually supplied may have been stored for quite some time, they may actually have poor viability. So there isn't that much of the live material in there. I mean, there's a bit of this in the literature where people have looked at lots of different products, and they've even found at times that there are strains, bacterial strains in the probiotic products that actually aren't on the label. And a lot of the selection of the strains that are out there at the moment have really been selected based on laboratory testing. So not testing actually in animals to start with it anyway, but on laboratory tests. But I think the biggest challenge to getting a probiotic that works really efficiently is that they have to work in quite a variable background. Within the animal, within the chicken, the gut microbiota can be really quite variable, and that means that there's a variable environment in which the probiotics are going, in which they have to work. And this is one of the big things that we're trying to address. So within the gut, you know, it's not just a uniform tube with the same bacteria, the same microbiota all the way down at the same functions. Obviously, in the different parts of the gut, there are different bacterial populations that are naturally resident there, and different parts of the gut obviously have very different functions. So the small intestine, that tends to be fairly similar in terms of the microbiota that's there. But it's very different to, you know, what's in the ceca. The ceca has a much more complicated microbiota, as does the upper part of the gut. So we know, various functions that are happening in the gut. We know the particular bacteria within the gut that have a role in some of these functions. And we want the probiotics to work within this complex system. So the key issue that we're trying to address with this product is really that combination of the variable gut environment. How do you get something that can reliably go in there and work? And our approach to that is to try and find strains that are highly persistent, that actually work as live organisms. They get into this variable gut environment, stick around and stay there, and so their action will be more reliable, is our hypothesis. And we should be able to do that without continuous dosing. So, our vision for this next generation of probiotics is that they'll be able to colonize the gut in the face of very variable microbiota. So, when feed is changing, the microbiota changes, whether it's a corn-based feed in North America or a wheat-based feed, in Australia, it should still work because it can actually get in there and stick around. And we think that ability to stick around will therefore give it much more reliable performance, and it'll have the added advantage of not having to dose continuously. Hopefully we'll only need to dose once or just a few limited number of times. But this requires the identification and development of bacterial isolates with these very special properties of being able to persist. So to find those very special bacteria that can act as these sort of probiotics, what we're doing is to only source bacteria from chickens. That's sort of a no-brainer, I would have thought, really. And we've abandoned the use of primary screening and laboratory tests, because even though this has been used for decades and there's a huge literature of people who have used it, and certainly some good probiotics have come from this system of screening in the lab, it's amazing, but when you go into the literature, there isn't actually a good case to be made. These laboratory tests are a good predictor of what will happen in the bird, which is what they're supposed to do. So we're actually trying to bypass the use of laboratory tests, and we're doing all our screening for probiotic strains actually in chickens. Now, the other point that's really important here is that it's quite counterintuitive, but we've found from quite a bit of work over many years that most bacteria that are isolated from a particular chicken gut can't reliably go in and colonize another chicken gut because the conditions are variable. It's got the problems with probiotics that I mentioned earlier, that if it's not tailored for that specific environment, then it's difficult to get in and recolonize. So this is a sort of quite unusual result. our work has shown that only about 3 to 5% of bacteria that you pull out of a chicken can really reliably go back in and persist there. So they're the special sort of bacteria that we're looking for, and this is the process we're using. So we're going into high performance birds, we're isolating lots and lots of different bacteria, then characterizing them, and then putting them into pools. And we put large pools of bacteria back into birds and then see which few bacteria we pull out. And these are our candidate probiotic bacteria. And then once we've got those persistent bacteria, we then go on to measure whether they've got some of those probiotic properties that I mentioned earlier. You know, can they improve gut integrity or the immune system? Do they kill pathogens? And then importantly, when you put them into animal trials, you know, can they actually give us some productivity improvements or you know, even just increase the flock uniformity by making the microbiota and hence how that's working more uniform in all birds. So that's what we're trying to do. So we're a good way down this pathway, and we're looking for these new probiotics for commercialization in industrial use. And in the final slide, you know, what we think will be the outcomes of this is that we'll have new generation probiotics that are specifically tailored for use in chickens, that can reliably be delivered to all flocks, irrespective of what sort of feed they're on, what the history is, what the environment is. And these will support health and productivity in all these flocks. And we should be able to do this with a single or just a few doses. And the hope then is that with the reliability and the lower dosage rate, we can get cost savings as well as more reliable benefits from these. So that's the project that we've got going. We hope that, you know, we're going to be able to supply the industry with these new probiotics relatively soon. And I'd just like to finish up by thanking AgriFutures for the support for this project. It's been really interesting and obviously can't be done without support from the industry funding body. So thanks.